stress and mainly focusing on salt stress and drought stress. And as you see, click. Okay, all right. So here's a map that I usually use on a lot of my slides representing all kinds of challenges that agriculture is facing. And you see a lot of the dots are blue dots, and blue dots are representing water problems. So we've got either too much water, too little water, or wrong kind of water, which is most of the time salt water. So my lab, we're focusing on drought and salt stress, so not enough water or wrong kind of water. And we are using resilient plants to do that. Arabidopsis is one of them. Arabidopsis is quite resilient, especially if you don't try to grow it in the lab. Then we're also focusing on close relative to cultivated tomato, Solanum pimpinellifolium, which you see over there. Its native range is west of South America, which is spanning quite a lot of different habitats. Some of them are quite extreme. Another plant that we're studying in the lab is tepary bean, which is native to southwestern US and northern Mexico, it has been used in by native people in their traditional agricultural approaches. So again, it's a natural resource for a lot of fabulous drought resilience trait. It's also very closely related to Phaseolus vulgaris, which is a common bean, which is famously resilient to transformation and tissue culture. Hopefully, with some of the approaches that Joyce is working on and Dan mentioned yesterday, we can kind of circumnavigate those challenges. The last but not least fabulous species that we're working on is cowpea. Cowpea is native to sub-Saharan Africa. Depending on which country you're actually talking to, they're saying cowpea is ours. It was domesticated here. But this is a super important crop for many of the smallholder farmers in Africa. It's a highly nutritious crop, but it's also super drought tolerant. And this is kind of flabbergasting because if you look at tepary bean, it has tiny, tiny leaves. So no wonder that you're drought tolerant if you've got very little surface to lose your water. Cowpea, on the other hand, has leaves as big as plates. Or as big as my hand. So I'm always really fascinated. How is this guy kind of figuring out, navigating those extreme drought tolerance? So, and in my lab, we're of course focusing on common drought or salt stress resilient traits like maintenance of the growth rate, transpiration rates, but I'm really interested in how stress is changing plant architecture. And we started with the root system architecture, which is quite easy to kind of like divide into specific components: main roots, lateral roots, the length of lateral roots, how the biomass is distributed. Another example of plant architecture is root-to-shoot ratio. We know that root-to-shoot ratio is extremely important under drought stress. We're not really sure what's happening under salt stress and what are the natural variation components that are contributing to those traits. Another thing that I really hoping to answer in the next couple of years is we know that wild plants have a variety of plant stature, or there's a difference in whether a plant is erect, prostrate, so growing like a carpet, or whining, and that's especially relevant for the beans. And those features, I think, are going to have tremendous effect on the xylem morphology and anatomy. And how is that kind of interplaying with its way of using water, maintaining the high water utility or high water use efficiency. So the tools that we're using are, there's a lot of phenotyping, and here are a couple of them. So we're describing those changes using sometimes high throughput, sometimes high dedication phenotyping. So of course we've got our BTI Phenosite, which is a high throughput phenotyping facility, which has opened this year. Yay! If you want to know more or get the tour, let me know. And we also have a little bit more humble tools like Raspberry Pi based pheno cages, or Spiro, which is for imaging plants growing on agar plates. And then once we are screening those natural diversity panels and we're validating them using, of course, CRISPR and stable transformation, what we heard from other talks, but we're also using Arabidopsis tDNA insertion mutants for the homologous genes, just because once Arabidopsis person, always Arabidopsis person. We also have recently adapted the protocols for virus-induced gene silencing in cowpea and tepary bean, and we're working on establishing the tomato hairy root transformation, which many people in here already have much more experience, so I'm always very glad and for all of their tips and tricks. So that's all. Thank you so much.